So today I will talk about cell cycle, cell division, mitosis and cell cycle and its regulation. So with this, let me share my screen. So screen ta visible ache? Is the screen visible?
Yes, sir.
Okay. So we'll talk about cell cycle. So each time a cell divides, there is a series of events series of দূর বাবা সিরিজের স্পেলিং ভুল হচ্ছে SERIES. There is a series of events which occur. So these series of events actually pass the cell from one point to another point, from one cell to two cells. And these is a series of events which a cell undergoes during division is known as cell cycle. So now if you look at a clock, if you look at this clock, this is known as the cell cycle clock. You will find that approximately 10 hours is passed in one phase. If you consider a clock of 24 hours, you will see that this clock is divided into some phases, some regions. First region is around 10 hours. The next region, say, is around 9 hours. Next region is around 4 hours. And there is one region which I am shading in black over here. This is around 1 hour. This region is around 1 hour. I've drawn it very poorly. Let me clean this drawing once more. So this region, this is 1 hour. This region is 1 hour. So this time point, this 10 hours, this is the known as the G1 phase, and this 9 hours is known as the S phase, and this G2, this phase is known as the G2 phase. Apart from that, this 1 hour is known as the actual M phase. So G1 you have S, G2 and M. So these are the 4 phases of a cell cycle you have. This M phase, G1 phase, is actually responsible for growth, metabolism occur during this time. The cell grows in size, it is getting prepared to divide. Then you have the S phase, where there is DNA duplication or DNA replication and chromosome DNA replication and chromosome duplication. You have DNA replication and chromosomes duplicate during this phase. Then you have the next phase which you have is known as the G2 phase. Cell prepares for mitosis. It checks whether the cell is in the position to go for mitosis. You have certain organelles disappearing, certain things, certain events happening during this G2 phase. And then you have the actual M phase or the mitosis phase which takes place. This can be divided into 2 phases. One is the real actual mitosis phase and the other phase is known as the cytokinesis. Here the cell materials during mitosis, you know that 2n it becomes into 4n and this 4n is again distributed into 2n and 2n respectively, and during this cytokinesis this is separated. So you have one cell actually having 2n number of chromosomes. During mitosis bivalents they become bivalents and tetrads, and it becomes actually 4n. It's actually 2n but 4c. So the DNA content duplicates and then it divides back into 2n, 2n, or you can say 2c, 2c. It had 2c present over here. 2c is the DNA content. So this is the event which takes place during mitosis. The actual mitosis takes place where this distribution takes place, and in cytokinesis the cell separates into 2. So this is this particular region till this whole region from here to here, that is these 3 stages together, they are known as the interphase. This is known as the interphase. G is also known as the gap phase. So it is the gap between the synthesis phase and the actual mitosis phase. In the M phase, you actually know the mitosis actually it can be divided into the stages: prophase, metaphase, anaphase, and telophase. So you know what the actual events which take place during prophase. During prophase, the cells condense, the chromosomes condense, cell organelles disappear, then all the chromosomes get attached to spindle fibers. In the metaphase, the spindle fibers, the bivalents, they go and stand in a single equatorial plate. In anaphase, the bivalents they separate. You have a V, U, I shaped chromosomes, they go towards the pole, and in telophase constriction starts to take place between those two separated halves, and then in cytokinesis the cells separate. So these are the things which you have studied right from your school days, all the phases of mitosis. I will not go through those into those details, although if you require then I can actually let us search for a picture of mitosis, and I can we can have a look at mitosis. So let's take this image. Are you able to see this picture?
Yes, sir.
Yes, sir.
Okay. So during the interphase, you can see that all the chromatid materials, they are all in a thin phase. This is the G1 actually. This is the the G1 S, G1 S, G2 taking place so in this region, and this is the actual M phase which is taking place in this region. So you find essentially the chromatids are condensed in this region. Then in the prophase you can see that these chromatid materials they start to condense. Remember, in the S phase you had one chromosome. This becomes chromosomes, they duplicate. This happened already in the S phase. So instead of having chromosomes like this, in the S phase the chromosomes have duplicated like this. The contents have doubled up. So they condense over here. In metaphase, then you can see here the spindle fibers are forming from the centrium cycle. They go to the opposite poles, they release spindle fibers, they get attached and they come to a plate. This is known as the metaphase plate. They come over here, and you have in anaphase they go towards two poles. See, they have the bivalents have become again more univalent, single chromatids again, and in telophase a constriction appears, the spindle fibers they disappear, the nucleus starts to form, organelles start to come, and in cytokinesis they separate out. So these are the essential events of mitosis. Now we will actually what we are going to talk about is events of cell cycle regulation. Now what causes the cell to pass from say G1 to S, and what causes the cell to pass from S to G2 to M, and from M back it goes back to again the next division to G1. What are the features which do this? So actually two key components have been identified. And what are these two key components? The first is a molecule known as cyclin, and the second is a molecule known as cyclin dependent kinases or CDK. Now a lot depends upon activity of cyclin depends a lot upon phosphorylation of this CDKs. So phosphorylation, actually phosphorylation say dephosphorylation, they are responsible for activation, deactivation of cyclin and CDKs. So these kinases actually they stay inactive, but when for example a specific cyclin comes and binds, CDK is inactive. Events of phosphorylation and dephosphorylation they take place and it becomes active. And what is its activity? It is a kinase, and the activity is actually phosphorylation of required components needed for cell cycle progression. So when a specific type of cyclin it goes and binds to a CDK, the CDK which was inactive, it becomes a CDK active, and what it does is it kinases phosphorylates, that is it acts a phosphate group, phosphorylates downstream target proteins which are needed to send the cell from one phase to another, say from G1 to S. So what is the basic philosophy of this? The basic philosophy of this particular thing is, it has been found that the levels of cyclin, they rise so these are the rise and fall during progression of a cell cycle. So say this is one cell cycle 1, cell cycle 2, cell cycle 3, like this, so and so it falls. So cyclin, these are levels of cyclin, they rise and fall. And it has been found interestingly that the MPF that the CDK activity they rise and fall at this time. This is a CDK. One such CDK is MPF activity. And what is its activity? The activity is phosphorylation, kinase activity. That is phosphorylation. This rises and falls. This suggests that the CDK cyclin, when they bind together, when the levels of cyclin falls, here the levels of cyclin falls, no CDK activity. Here it rises, you can see high CDK activity. And CDK activity, so the rise and fall of cyclin is a very important phase which regulates the cell cycle. Rise and fall of respective cyclin. There are different types of cyclins for different phases of cyclin is very important for CDK activity and eventual cell cycle progression. Now there are essentially, according to Alberts et al., Albert's book, there are three types of cyclins. The first is the G1 S cyclins. The second is the S cyclins, and the third is the M cyclins. And in what in certain a fourth class is sometimes present, and it is known as G1 cyclin. This is a G1S cyclin. This is the S cyclin, M cyclin, and a G1S cyclin is present. So what does a G1S cyclin do? So G1S cyclins, they activates CDK in late G1 and thereby trigger progression to start there is a start. Okay, start progression resulting in a commitment for cell cycle entry. Okay, now start G1 cyclin. Then what does the S cyclin do? What do they bind to CDKs soon after progression through start, when the start of the cell cycle takes place. Through start and help stimulate chromosome duplication. It is interesting to note that S cyclin levels remain elevated or high until mitosis. So we are talking about 3 to 4 types of cyclins. Then you have the M cyclin. They are also known as the mitotic cyclin. MPF. metaphase promoting factor. Stimulate entry into, some people call this also as mitosis promoting factor, stimulate entry into mitosis. So they promote the entry from G2 phase to mitosis. So the fourth class of the G, then we had talked about a fourth class of cyclin. That is the G1 cyclins. So the G1 cyclin are the fourth class of cyclin. They help growth of cells. Growth of cells. That is, they work during the start of G1 phase. So what are the different types of cyclin and their respective complexes? Let us have a look at of them in yeast. So what is the name of the cyclin? CDK complex. Then this is in vertebrates we are talking about, and the cyclin in, sorry, not in yeast, this is in vertebrates. In yeast also there are ones. I would think that it's better you study about the vertebrates. And the name of the CDK, the first is the G1 CDK complex. Name of the cyclin is cyclin D, CDK4. Then you have G1S CDK. This is known as cyclin E, and the CDK is CDK2, cyclin dependent kinase 2. Then you have the S CDK. This is A. This is also CDK2. Then you have M CDK, and the name of the cyclin is B and CDK1. Now let's have a look at the, let's draw the picture. G1 S G2, then let's draw M. Again after that G1 starts. So let us talk about the G1S cyclin. Starts from here, the level rises and it falls when S phase starts. It falls here. and it again starts to rise. This is the G1 S cycline. Next we talk about the S cycline. Oh, this is also the symbol, the signal for start or go, to start, and the cycline which is formed is G1S CDK. At this point, what works is the SCDK complex. So the levels are low now. It increases here, stays throughout here till the middle of mitosis, and it goes away. It again falls and then it starts to rise again. So this is the level of the S cycline. We are talking about cycline which cycles, not the CDK. The CDK is bound over below. We are talking of the CDK is below. Then you have the third group of cycline, the M cyclines. From the G2 phase they start to increase. They start to increase here. And their levels fall here. So these are the M cyclines. And G2 phase is controlled by MCDK or the G2 M cycline, G2 to M. And at this point there is another compound which works. This is during this phase. Let me draw it properly here. Let's draw at blue. This is known as the APCC CDK20 complex. This is the anaphase promoting complex. Also note that all these three things, this one, and this one, they are known as the critical checkpoints. So now what are cell cycle checkpoints? We will talk about. Ki holo mama? Talk about cell cycle checkpoints. So we saw that there are three checkpoints. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. 1, 2, 3. The first one was the G1 by S checkpoint. The second is G2 to M checkpoint. And the third is metaphase to anaphase checkpoint, or this is known as the APC by C checkpoint. These two are known as the DNA checkpoint, and this is known as the spindle checkpoint. So, what is the importance of these checkpoints? The G1S checkpoint it's better if I draw it with a figure. So, say this part to this part is the S phase. This part to this part is the G1 phase. This is the G2 phase. And this is the M phase. So, you have a checkpoint over here. G1 to S checkpoint. You have another checkpoint over here. And you have in between another checkpoint, APC by C checkpoint. So there are three checkpoints which you see. This checkpoint, it ensures cell has grown enough and is ready for cell division. So this point suggests that the cell is grown enough, and now since cell division or it's a cell for DNA replication, rather it's a better word, DNA replication. This ensures that the cell is ready for DNA replication. DNA replication is a very expensive process of the cell. It is very expensive process for the cell, and because of this expensive process, the cell must ensure that it is in the perfect condition to start replication. Any error in replication will either kill the cell, or you have some mutations or certain defects which are detrimental. So this is not allowed. Then we have the G2 to M checkpoint. They will ensure whether the DNA has been, S phase has gone correctly, whether DNA has ensured sufficient proper replication. If there is a proper replication has been ensured, only then this phase will go ahead. And APC spindle checkpoint, If all the chromosomes have correctly attached to the metaphase plate, they are lying perfectly in the equatorial zone. If they are not lying, you will see that if one side you'll have Turner syndrome and Klein syndrome type. One side you'll have more chromosomes, one side you have less chromosomes coming up. So these are all developmental defects which take place in the cell, which is not wanted. So there are three checkpoints. If these three checkpoints, if they pass through these checkpoints, only then the cell will perfectly divide. Otherwise the cell will die, or it will go back to G1. It will go to apoptosis, to death. So there is a very strong regulation taking place. Okay, so let us now look at the whole events of the steps of regulation. Step by step, let us look at the steps of regulation of cell cycle. I will write this down very slowly. You people can go through it very carefully. Steps of regulation of cell cycle. So what happens in the first step? Rising levels of G1 cyclins. When the cell starts, G1 cyclin, they bind to respective cyclin dependent kinases. In this case, if you look at the figure, it is CDK4 cyclin D, and signal the cell to prepare chromosome for replication. Signal the cell to prepare chromosome for replication. So during this phase, if you remember the picture, a rising level of S cyclin what were the S phase cyclin? If you see clearly, E and A in the table, and their respective CDKs. CDK2, CDK2, they bind so cyclin E and A bind, so you have two of them forming CDK2 cyclin E, CDK2 cyclin A. It is formed. I keep them in brackets over here for you to understand later. And their subsequent entry into the nucleus. So all these activities were taking place in the cytoplasm, so they now enter into the nucleus. So what it does is, this prepares the cell to duplicate its DNA, also centrosome. So now the DNA replication has started. As DNA replication continues, cyclin EA level, which binds to CDK2, levels go down, and the levels of mitotic cyclins begins to rise. In the G2. So what are these things? Cyclin B, CDK1 complex. This is also known as the MPF complex. In the next step, there is translocation of, this happens in the cytoplasm. Now it again goes into the nucleus. There is translocation of cyclin B, CDK1 by MPF, whatever you call them, into the nucleus. And this initiates following thing. First thing, now mitosis is starting. From G2 to M we are going. First thing which is as the assembly of the mitotic spindle. Spindle fibers start to form because of this cyclin B, CDK, MPF, because of this forming. Assembly of mitotic spindle. The next step, what is happened? The nuclear envelope breaks down. The nucleus is breaking down, disappearing. Breakdown of nuclear envelope. The next is stopping of transcription. All transcriptional activities which are taking place in the DNA, they stop. They have taken place in the G1 phase. All transcriptional activities had taken place in the G1 phase. That is where metabolism, transcription and all they have taken place. In the interphase, all of them. In the metaphase, everything stops. All organelles, they disappear. stopping of transcription. And you know the next thing which what happens is condensation of chromosome which takes place. Let us see the next step. So all these events which have taken place here, these events take the cell to the metaphase, to the cell to the metaphase stage of mitosis. Now let's go to the sixth point after this. Now at metaphase stage, at this point, the MPF activates the APC by C complex. That is the third checkpoint. Activates the APC by C complex, which allows the sister chromatids at metaphase to separate. Which allows the sister chromatids. I will talk into details about the mechanism, but this is the overview. Sister chromatids at metaphase to separate. So actually what happens, the problem while you are reading it is you forget what are the sequences which happen, what happens after what. So I have just listed down, in no book it is written, I have just listed down what happens after what. So this is going to help you to study, get an overview about the whole of this mitosis. To separate. So what happens, you know that they are attached to one another. There are molecules known as cohesins. So this APC by C, after what it does, it allows these to separate. So from metaphase they move to the anaphase. Now here comes the real question. For separation, so all these chromosomes, they are bound by molecules known as cohesin. This cohesin, this needs to be dissolved, and this allows separation of chromosomes. So for separation, cohesin needs to be broken down. This cohesin molecules needs to be broken down. So breakdown is caused by a protease called separase. So separase breaks down. So if this is breaks down cohesin. Separase also is known as separin. So separin is normally kept inactive until late metaphase, when anaphase wants to start, by another compound. Separase is blocked by another compound or another molecule, a chaperone, known as securin. This is a chaperone. Keeps separin inactive till anaphase starts. So now when anaphase starts, let's go to the 8th point. Anaphase begins when APC by C complex destroys securin. So anaphase promoting complex by C, what it does? It destroys securin. What happens when securin is destroyed? Separin is active. What it does? Cohesin is dissolved. And what it does when cohesin is dissolved? Chromosomes separate. Then APC by C complex, it destroys MPF. Maturation promoting factor, mitosis promoting factor, metaphase promoting factor, destroys MPF complex. By destroying the B cyclin. Then the cyclin level falls down because it's been destroyed. CDK levels are constant, but it's active and inactive. But cyclin levels rise and fall. It is falling because the B cyclin of MPF is being destroyed by APCC. So anaphase is promoted. So this ends my class on cell cycle regulation. In the next class I will be talking about in details on certain events which take place during mitosis. তোমাদের কারোর কোনো কোয়েশ্চেন আছে?
না স্যার, ক্লিয়ার আছে।
ক্লিয়ার আছে তো? আমি রেকর্ডিংটা পাঠিয়ে দেবো, বা অসুবিধা হলে জিজ্ঞেস করে তোমাদের নোটগুলো দেখো, সকালে হোয়াটসঅ্যাপে পাঠিয়ে দিয়েছি। ওকে ওকে, দেন রাখছি তাহলে, আমি তোমাদের নেক্সট ক্লাসটা বলে দিচ্ছি। ঠিক আছে, ঠিক আছে।